Okay, good morning all. Today we are going to start next topic and it is food microbiology. Food microbiology basically microbes present in the food and how we can use the knowledge of microbiology to preserve the food, to save the food, we will utilize it and we can preserve the food for the longer duration. So in this topic we will see these are all things and first we will see the basics of the food microbiology, then we will go for the advanced. Now food production is a very specific area and we know nowadays many bigger multinational companies is there in the food production, and these multinational companies they are preparing our packaged food, and these packaged food we are utilizing for our daily needs. So food there are therefore has to collect and store of use of during period of low and no food production, no food number of food production. So food, what we are doing, food production, if we are talking, means collection of the food from the farmers, and we are storing for the longer duration, so that we can utilize those food material for the longer duration time. So food storage is complicated by the fact that food begin to deteriorate shortly after harvest and gather or slaughter. So when we are collecting the food material from the farmer, our target to utilize those food material for the uh, feeding purpose. Okay, if the feeding time is late, if you have to feed the people after the 1 week or 15 days, then you have to utilize the storage technique, so that we can increase the timing of the storage, and then we can utilize after the 15 days or 1 month also. So what technique we have to utilize so that food timing, food storage timing we can increase and food will not be waste. So these are all things we will study in this topic. Now first we will have to understand what is food spoilage. Food spoilage is a damaging or injury of the food. If food is damaged, if food is very, food is not in the natural condition, how we gather when we are collected from the farmers or the grower or any shop which is producing food. So if it is not in the good condition, if it is the microbes is attacked or fungi is spoiling the food, so that food is spoiled. Means food is not suitable for consumption. So if we will consume, then we will be in big trouble. Maybe food poisoning we will have, maybe we will be ill, or maybe we have some other disease, because when microbes, pathogenic microbes is there on the food, if we will consume, so those pathogenic microbes also cause some other disease. Okay, and we have some other disease. So food must be considered spoiled if it is contaminated with the pathogenic microorganism and non-pathogenic microorganism and some poison also, if it is there, pesticides, heavy metals. If these are all things, if it is there in the food, it means the food is contaminated or food is spoiled, food is waste, food is not suitable for the human consumption. Okay, now storage life of some food. See, different food is there and they are having natural capacity to cope up with these food spoilage. After that particular time, they will can't able to store it, because the timing between the food storage and utilization, it will be finished, and after that it will start the spoiling, and it is not usable for the consumption. That's why if see these are all food, and if more than of the storage life is passed away, then you can't able to utilize because it will spoil. So if you will see the raw beef or mutton is there, so 1 to 2 days. Raw fish and raw poultry product, if it is there, 1 to 2 days you can utilize at the temperature of 21 degree. If you will increase the temperature, if suppose 37, then 1 to 2 degree will 1 to 2 days we can't able to store in the open. Only it will be there for some hours, 12 hours like that. because 37 degrees is more suitable temperature for the bacterial group. Okay, and 28 degree for fungus group. So if you will keep those food item on the optimum temperature, then bacteria and fungi will grow very fast. Now these dried, salted and smoked meat and fish, when we are adding salt inside the this raw food product, so we can increase the timing of the storage. So see where dried, salted and smoked meat or fish, if you have, you can keep 360 or more degree, because we are adding salt, so that shelf life or storage life will increase. Fresh fruit you can store 1 to 7 days. Dried fruit 360 or more days, because it is dried completely. There is no water. If water is not there, then microbes, fungi, they will not attack. Enzyme will not be utilized without water. So dried, any type of dried fruit you can store more number of days. See, dried powder of the many vegetables is nowadays available. Dried fruit is available from the starting itself, and many dried spices. These spices we can store because they are dried. So these dried powder we can do, or dried fruit we can utilize, or we can store more days. Okay. Then leafy vegetable 1 to 2 or 3 days. Mostly it will be you can store it. Then root crop 1 to 20 days. Dried seeds 362 or more. So here is two type of things you saw: the dried thing and no dried thing. The water content, if it is more, then you can have the less number of days you can store it. If water content, if there is no water content, then you can increase the number of days for storage. See, the dried fruit is there and dried powder of the other food material you can store more days. If water content is there, like leafy, root, fresh, vegetables and poultry products, fish, beef, mutton, so storage time is very less. So water is a main thing. Like water is present, then bacteria and the fungi, they can release the enzyme and they can utilize the nutrition from the food item. In most cases, there does not need to an evident sign of spoilage, and by seeing itself, can't able to see say that the food is spoiled, and it might look good or normal. But even though if you will eat, it will be like good. So that microbe, they are just spoiling just starting the spoiling, and you can't able to see that these bacteria are releasing or digesting the food material, and they are releasing toxin. So by bacteriological and toxicological investigation only we can able to do the we can able to say that the food material is spoiled. So when any food is start spoiling, at that time we can't able to just see and just taste. We can't able to recognize that food is spoiling. So we have to do bacteriological and toxicological investigation, so that we will able to know that bacteria is spoiled. When food decay, when time is passed, sometime use 1 day and 2 days, then only you will see the changes in the food color, taste, odor is there. So these things you can able to visualize by your eyes, and you can smell it, and you can say that food is decayed and the food is spoiled. Now, why food is spoiled? What is the reason of food is spoiled? So first one is the growth and activity of the microorganism, because the bacteria, fungi, and protozoa, they are all growing inside the food, and they are the main reason because the food is spoiled. When any microorganism is growing on the food, they will release the enzymes, and they will utilize the nutritious content of the food for their energy and growth and the development, for their energy production as well as for the growth and the development. So ultimately, for their growth and development, they require nutrition. That's why their needs, because they need nutrition, so they are attacking on the food items. So they produce various enzymes, and that decompose the various constituent of the food material. Now, enzyme actions is there. Many enzymes also present in the food item. The natural plants material, fruits, vegetables, and meat. They are also having their own enzyme. They are the part of their cells. Means our cells also producing enzymes, animal cells also producing enzymes, plant cells they are also producing enzymes. So when these enzymes is part of the cells and present in the plant and animal tissues, and they will start the decomposition, and they will start the decomposition means it will take some time, means 2 to 3 days it will take to decompose it. So they, if we are not preserving, so automatically they will start decomposing. Then many chemicals we also know is present in the cells, and these chemicals also will react with each other, and they will react with the enzyme, and they will do the food spoilage. Example is oxidation of fat. So food fat oxidation can is a type of chemical reaction. So we know the many type of fat is present in the cells of the plant and animal, and these fat molecules can be oxidized, and then when it will be oxidized, so a taste will be changed, the order will be there, and these order and taste we are calling food spoilage. So chemical reaction, enzyme activity, growth, and microorganisms, these are all the main reason of the food spoilage. Other reasons also there, like vermin. Vermin means we will on rats, cockroaches, mice, birds, larval stages of some insect. They are all we are calling vermins. If somehow birds or cockroaches or rat are came or they are spoiling the food. So these vermin also spoiling the food. So, as aesthetic aspect of the presence, possible transmission of pathogenic agent, and consumption of the food is the main reason these are all vermins will be present in the food material. Because these vermins means cockroaches, they also require food material. So they are coming and eating and they are spoiling the food material like birds, mice, larvae. So these are all vermins are for their requirement of the energy, they are eating the food material and they are spoiling. Now physical changes also there. When we are keeping the food material will be physically changed, and we can it will be spoiled. So we can able to when we are keeping in the freezer environment, like we are reducing the temperature, or maybe we are increasing the temperature, so that microbes will not be spoiled. But when we are increasing or reducing the temperature, physical characteristic of the food will change. Physical characteristic of the food will change. And during cooking, when we are cooking the food, so we have to give the heat. So when we will give the heat, physical characteristics of the cells will change. And when the physical characteristic will change, so it will be more prone to spoil fast. So these are all the reasons is there. Now microbes in food spoilage. So many microbes is there like bacteria, yeast, and molds. They are the major food spoilage region. They are the major causative agent of the food spoilage. They produce various enzymes, and these enzymes only digest the food material. Okay, so these bacteria, yeast, and mold, what they will do? They will release the enzyme to digest the food material, and because the food material is digested, so it is not suitable for the consumption purpose. Okay, molds, they are the major cause of food spoilage, and they will reduce the water activity, dry cereals, and dry products. Even though if your dry cereals and cereal product if they are having water in limited amount, so these molds can be utilized those cereals and their products, and they will spoil the food. Bacteria spoil food with relatively high water activity, such as milk and butter. So molds require less amount of water. If less amount of water is present on the food, molds will spoil the food. If bacteria, if you will see, bacteria need more water content in the food. More water content, so bacteria will attack easily and they will spoil the food. Then sources of microorganism in food. So what sources is there of microorganism contamination, how these microorganism is contaminating the food item. So source, many source we know, first is soil and water, plant and plant products, food utensil, intestinal tract of man and animal, food handlers is there, animal hides and skin, air and dust. So these are all the different things is there where these microorganism is coming through these medium, and they are, after the entering inside or taking to the food, they will spoil completely food material and food will be not suitable for consumption. Now, factor affecting microbial growth in the food. Two type of factors is there. Intrinsic factor, the factor which is inside the food, internal part of the food. So these factors, many type of factors there, like hydrogen ion concentration, pH, moisture content, nutrient content of the food, antimicrobial substances, biological structure. One by one we will see. Then second one type of factor is extrinsic factor. Extrinsic factor is the factor which is not related with food, like environmental factor, temperature is there, humidity is there. So these intrinsic and extrinsic factor we will discuss in detail. First one is hydrogen ion concentration or pH. We know that broad range of pH we are utilizing on the food. Means some food material is low pH, uh, acidic pH you can say, some pH, food material which is having neutral pH, and some food material which is having alkaline pH. So these food material which is having different pH range, and these pH range can attract the mold and bacteria, because many bacteria can able to grow in the acidic environment, like salmonella is there. Many food, many microorganisms which can able to grow in the alkaline environment. So alkylophilic bacteria is there because they are like alkaline environment, and many molds and fungi they like neutral type of environment. So neutral pH they require. So different pH is required by different microorganism, and these microorganisms can spoil the food material. Now food as an example we will see what type of food is having what type of pH range. See, beef is having 5.1 to 6.2 pH, chicken is 6.2 to 6.4, milk 6.3 to 6.8. So these are all basically acidic environment. Cheese, fish, oyster, milk, chicken, beef, they are all having acidic environment. See, many fruit varieties like citrus fruit, especially they are more acidic, less than 4.5 and most less than 3.5 is there, and vegetables 3.0, 6.1. Then these are all acidic environment, the type of food. So most of the food material we are eating, they are all acidic environment or acidic pH. And many bacteria they can easily utilize these food products because they are having capacity to grow in the low pH. So acidophilic microorganism they like the acidic environment, and many food material they can able to, they have the acidic environment. So that's why these acidophilic microorganism can spoil the food material. Okay, so yeast molds grow under acid condition. Many molds and yeast they will grow under the acidic condition. So these are all food can be easily destroyed by the yeast and molds. Then Vibrio cholera are very sensitive to acid, and they like alkaline environment or alkaline pH. So alkaline pH type of bacteria is there. They like the alkaline pH. So most of the bacteria are killed in strong acid or a strong alkaline environment, except the microbacteria and their different species, they don't have any effect on the alkaline and acidic environment. They will live without any problem, and they will spoil the food. Okay, the different species there, some are pathogenic, highly pathogenic, some are non-pathogenic, and they are spoiling the food material, and without pH they are not affected. A minimum and a maximum pH for growth of some specific microorganism. So E. coli required minimum 4.4 pH and maximum 9. So in between 4.4 to 9, E. coli can able to grow. 4.5 to 8.8, Salmonella typhi can able to grow. Then mostly all of the bacteria can able to grow 4 to 9 pH, in between 4 to 9 pH. Bolts can able to grow 1.5 to 11 pH. Yeast can able to grow 1.5 to 8.5. So the suitable pH is 4 to 9. If the food material is having 4 to 9 pH, the most of the bacteria, molds, and yeast can able to grow easily on these type of food material. Next is moisture content. Moisture contains the amount of water present. How much of amount, or actually free type of water, the freely available water. The water which is bonded with the chemicals, we are not counting, that is not free because it is bonded with chemicals, so it is not free water. So only free water, the amount of free water we are calling the moisture content. And that free water only is important for the growth of the microorganism. If food is having more free water, if food is having more free water, then the microorganism will grow very easily. If food is having very less water, then the chances is there food spoilage will be less. If there is no free water, like your dry fruits is there, nuts. So many nuts they don't have water, or very less content of water is there, free water. That's why their shelf life is 360 or more than that. 360 days. If there is lack of free water, microorganism will not grow. So nutrient content of the food, like how much protein is there, carbohydrate is there, lipid, water, energy, nitrogen, sulfur, phosphorus, vitamin, and mineral is present in the food material. If it is very rich in the food material, but there is no water content, then microorganism will not attack, because for the activity of enzyme, for the activity of microorganism, water is very essential. If water is not there, microbes will not attack. So that's why dry powder, dry vegetables, and dry nuts, 100% dry nuts, you can store up to 360 degree in the open environment. In the shops, when you are going to purchase these type of food material, they are keeping in the open, and no spoilage is there, because bacteria and fungi, they will not attack on the food material which is 100% dry. Right. Okay. So foods such as milk, meat, egg contain a number of nutrients, and if water content, free water is also available, these microbes will attack and they will spoil the. Now, antimicrobial substance. Many food material, they are having chemicals, and those chemicals can inhibit the can able to inhibit the microbes and molds and other microorganisms. And these and chemicals which is working as an antimicrobial, the chemical which is killing or inhibiting the microbial growth, and they are natural part of. Then biological structure. Some food have biological structure that prevent the microbial entry, and we know there is meat has fascia, skin, and other membrane and prevent the microbial entry. And eggs is having shell. So if shell is there, so microbes can't able to attack. So natural some barrier if it is there on the food material, like your seeds. Seeds is also we are utilizing for food purpose, and all seeds are having seed coat, and these seeds coat can able to prevent the microbial spoilage and entry, and microbial spoilage and entry can able to preserve the food material. So biological structure is there so that food can be stored longer duration. Then extrinsic factors. These are the external factors, and they are affecting the microbial growth, and temperature is there for storage, presence of concentration of gases in the environment, relative humidity. These three are we will discuss. So first one is the temperature. We know that microbes required a particular temperature for the food material on the will be there. If we are reducing the and keeping the food material in the low temperature, if we are increasing the temperature for the storage purpose, then we can able to easily preserve the food material. Suppose the food material we are stored in the zero and sub-zero temperature, so we can store nicely, and the life of the food or spoilage free food we can able to preserve in the longer duration. So temperature is very effective factor. We can stop the growth of the microorganism when we are keeping in the low temperature. And in the low temperature, the food characteristic will not be degrade. If you are keeping in the high temperature, food will be damaged because of the high temperature. And enzyme and other chemical constituent of the food, they will change. That's why the high temperature is not preferable, but low temperature or very low temperature like -10, -22 degree centigrade. If you will keep these food material, they will be automatically preserved for the spoilage. Then psychrophilic microorganisms, you see some microorganisms, they are extremophilic. They can able to grow in the low temperature also, like 0 degree, -10 degree. So 0 to -10 degree, they can able to grow very easily. So these extremophilic bacteria, they can able to utilize these food material, and they will spoil the food. And several microorganisms found in the soil and water belong to these psychrophilic microorganisms. And these psychrophilic microorganisms, whenever they will able to infect or they will able to contaminate the food, the food will be spoiled in the low temperature also. Then next is mesophilic bacteria, and these mesophilic bacteria can able to grow in the normal temperature like 25 to 40 degree, and for their growth the optimum temperature is 37 degree. And many pathogenic and non-pathogenic type of bacteria is mesophilic type of bacteria. They will grow in between 25 to 40 degree, and 37 degree we already know is the optimum temperature for the bacteria. So if we will keep our food material in between 5 to 43 degree temperature, then food will be spoiled very fast. Okay, so if we are keeping less than 4 degree, and if we are keeping the food material more than 45 degree, so that we can able to increase the storage time easily. Okay, so most pathogenic bacteria belong to these type of group, and most of the pathogenic and non-pathogenic, and many non-pathogenic bacteria also belong to mesophilic type of bacteria. Now thermophilic bacteria, thermophilic, they like the high temperature. Thermophilic bacteria, they like the high temperature, and because they like the high temperature means they can grow on the high temperature. So the bacteria can able to spoil the food material on the high temperature more than 45 or 50 or 60 degree. So if the food material we will keep on 40 45 or up to 70, these bacteria can able to spoil the food material. So many bacteria is there, they can able on the high temperature, like and they are thermophilic bacteria. And most of these bacteria are spore forming bacteria, because in adverse condition, if temperature will go up to 75 or 80 degree, they will form the spores. Then concentration of gases in the environment. If the concentration of gases is changed, because many aerobic bacteria required oxygen, and non-aerobic bacteria they don't require oxygen. If oxygen is not there for aerobic bacteria, then growth of the aerobic bacteria will be reduced. If oxygen is there and anaerobic bacteria is there, so growth of the bacteria also reduced. So gases concentration is very important, because aerobic bacteria required oxygen, anaerobic bacteria don't require any oxygen. So if concentration is reduced for the aerobic bacteria, and concentration of oxygen if we are providing for an anaerobic condition type of bacteria, then it will not be good condition for the growth of the bacteria, and they will not spoil the food. So the oxygen concentration, carbon dioxide concentration is very important when we are talking about the food spoilage, and we can decrease the oxygen concentration, and we can be able to increase the food storage time. Now food affected by various groups. So many food various group is there like anaerobic, aerobic, facultative bacteria, micro aerophilic bacteria, aerobic molds. These are all different groups of bacteria is affected or spoiling the food. So first is anaerobic bacteria or facultative anaerobic. They don't require any amount of oxygen. They require only carbon dioxide environment. So they are the spore former and most likely to grow in the canned food because canned food is totally packed. So packed food material, the oxygen will not penetrate, so they don't require oxygen. That's why they like to grow inside the canned type of food or inside the closed environment. So microaerophilic bacteria, they likely to grow in the vacuum packed food since they have low oxygen demand. Then aerobic bacteria they grow in the open type of environment because they require more amount of oxygen and many type of food material is spoiled because we are keeping food material in the open environment. So these aerobic type of bacteria they can able to grow and spoil the food material. Then aerobic molds is there will grow in the insufficiently dried or salted product. So aerobic molds, they will grow if the your dried product is insufficiently dried. Some water content, if it is there, so these water content is promote the aerobic type of mold growth. These aerobic type of mold growth can able to grow very easily and they will spoil the food material. Then relative humidity. We know the if environment, if air is having water content, so our dry food material they will absorb the food water, and when they will absorb the water content, so that microbes they will attack very easily and they will spoil the food material. So environment relative humidity, if it is there in the environment, the food material will absorb the water from the environment, and when they will absorb the water from the environment, they will get easily spoiled. So always your humidity will be less in the environment so that you can preserve the food from the longer duration. Now, food preservation. So, food preservation itself is a area or a subject, and there we how we can able to preserve the food we will see, and for the longer duration, if we can able to preserve the food so that we can able to utilize food material for the longer time. So, food preservation is a process which physical or chemical agent we can utilize to preserve the microorganism spoilage of the food material. So food preservation aim at treating food in a manner to prolong its storage life. In food preservation, efforts are made to destroy organisms in the food and increase the period taken by the microorganism to adapt the food environment before they start to spoil of the food. So what we are doing in the food material, we are preserving the food material against the growth of the microbial spoil. So we are giving the physical and chemical treatment. So these physical and chemical treatment can able to preserve the food material. So food preservation principle is depend on the two type of two type of food preservation. First is inhibition principle. Second one is killing principle. So two type of things we are doing: killing of the microbes or inhibition of the microbes. So first we will see the inhibition of microorganism. So inhibition principle is depend on the food preservation. Food preservation is achieved by the inhibition of growth of and multiplication of the microorganism. So growth and multiplication of microorganism we will inhibit. So automatically we can able to preserve the food for the longer duration. So this we can achieve by the reduce the water content of the food. If we are able to remove the water content of the food, then food will be stored for the longer duration. If we are keep changing the pH, if we are changing the pH for the food material, if we are reducing or if we are increasing the pH, so that microorganism will not attack and the food will be preserved. Then use of preservative like some chemical we will add, and that chemical can able to preserve the food material against the microbial growth. So these chemical we can use as a preservative like sodium benzoate is very chemical name sodium benzoate, and we are nowadays adding in the food material, and this will preserve the food material for longer duration. Then use of low temperature, we can keep the food material in the low temperature so that microbes will not produce any enzyme and they will not inhibit the food item, or they will not spoil the food item. Then smoking, which has the drying and preservative effect. So when we are doing the smoking, so food will be preserved or dry, and they will be stored in the longer duration. Then inhibition methods, preservation of the food by inhibiting method does not necessarily imply the destruction of the organism. Just we are inhibiting the growth. We are not destroying the organism. So organism will be there, but just we are inhibiting the growth and development so that they will not release the enzyme, they will not grow, and they will not destroy the spoilage. And we can keep it on the inhibition of the microorganism for the some time so that we can utilize those food products. Now food preservation by lowering pH. So we can decrease the pH of the many food products so that we can increase the storage time of the food material. So lowering of the food material pH can be achieved by the addition of acid and fermentation. So we can add the acid either in the food, or if we will ferment, so acidic environment will be produced, and these acidic environment can stop the microbial growth. So from the changing the pH, we can able to store the food material for longer duration. Now food preservation by lowering water activity. If suppose the free water, if you will decrease in the food material, so the microorganism can't able to attack, and we can able to preserve the food material for longer duration. So addition of high salt we can add so that we can decrease the water content. Sodium chloride and sometimes so nitrates and nitrites also we can add so that water activity will be reduced and the salt concentration can able to increase. So the salt increase the salt concentration can able to preserve the food material. Then addition of high content of the sugar also will be same effect how we are increasing the salt concentration. Then drying, sun air dried or electric dried or freeze dried product of the food material, we are reducing the free water so that the water content will be reduced in the food material and microorganisms cannot able to attack, and when they can't able to attack, so we can increase the food storage time. Desalting procedure. Desalting procedure can be performed in the four-way. The dry cure, in which the meat or fish is rubbed with the salt. Pickling, the product are members in pickle of brine, usually containing about 15% salt. The injection cure, concentrated salt injected into the muscles. And fourth one is direct addition method is there. So if we will rub the salt on the surface of the any food product, then it will be pure, because outside only microorganism it will attack, then only we can able to preserve. Pickling is a process where we are making pickles, and these pickles usually we are preserving in 15% salt concentration. So sometime what we will do, we will inject these all salt in the liquid into the food material, so that when we use these injections, so we are calling injection method. Then preservation of food by addition of high content of the sugar. So many sugar like glucose, fructose, sucrose, we will increase the concentration in the food so that the microbes can't able to utilize those sugar content, because the high concentration of sugar, especially glucose, fructose, and sucrose, can able to decrease the activity of the microorganism, and the food material will be preserved. Thermophilic are more susceptible to the action of sugar than other bacteria, and osmophilic yeasts are able to tolerate very high concentration of sugar and cause food spoilage. So some yeast species they are able to tolerate high concentration of sugar, and they are basically high concentrated sugar content of the food material. These yeast will spoil very easily, and they are the main reason of the food sugar content food spoilage. Then food preservation by use of low temperature. Any food material we can store in the very low temperature, or we can store in the cold storage, and these freezing temperature we can keep up to 0 degree to minus 10, 15 degrees, and chilling is keeping food at low temperature between 0 to 15 degrees centigrade, and the common chilling temperature range is between 4 to 5 degrees. So freezing is keeping food at temperature between 0 to minus 35 degree, and this chilling effect enable food material for the longer duration. Now, effect of low temperature. When we are keeping the food material for the low temperature, these low temperature can able to preserve the food material, because many enzymes when we are keeping in the low temperature they will not be functional. Enzyme also required a perfect temperature for the conversion of substrate to product. If that temperature is not there, then enzyme will not be substrate to product formation. So a low enough temperature will prevent growth of any microorganism. Spores are not usually injured by all freezing type of temperature. However, most parasites are killed by the freezing or low temperature. In clean principle, microorganisms are destroyed in the food, and the food protected against subsequent contamination by being enclosed in airtight container. Now, method employed to achieve the killing principle. First one, heat treatment to pasteurization or heat sterilization. We can increase the storage timing. Irradiation, we can use ionizing and non-ionizing type of radiation. And these radiation can able to reduce the uh, microorganism, can able to kill the microorganism 100%, and these radiation can able to increase the storage time of the microorganism. Then uses of gas. So ethylene oxide or ozone, these two gases is the can able to destroy both the vegetative cells and spores. And when food material will be in close contact with the ethylene oxide or ozone, these gases can able to kill effectively the vegetative cells and spores, and it will be help in the preservation of the food. Next method is pasteurization. So pasteurization is the process where heat treatment we are providing to the specific temperature and times, and then we will treat with these food material on the specific temperature and time the food will be preserved. So pasteurization is aimed at destroying all pathogenic microorganisms without affecting the nutritive value of the food. Three methods of pasteurization we know is there. First one is the low temperature for the low time, and in this method 63 degrees centigrade for 30 minutes we will utilize for the pasteurization. Then second method is high temperature for short time. So 72 degrees centigrade for 15 seconds we can keep the food product and they will be pasteurized. Then third method is flush method, and we are increased temperature 80 degree for 1 to 2 seconds, and these three methods we can utilize for the pasteurization of the food. And sterilization, in this when we are utilizing physical or chemical methods to destroying the microorganism, and it will preserve the food material. So sterilization can be achieved by the heating at the high temperature like 100 to 140 degrees centigrade, and irradiation kill the bacteria, spores, and insect, as well as inactivities of the enzymes also can be achieved when we are increasing the temperature. Now applications. In practice, often a combination of inhibition and killing principle and the various methods are used depending on the food type, like use of pasteurization and chilling of milk, lowering of water activity and low temperature storage, use of preservative and low temperature. So these are all the various applications there for the food preservation, so that food will be preserved and we can able to utilize for the longer duration. Now, three important terminologies we are utilizing when we are using heat in the food preservation. First one is D value, second one is Z value, and third one is F values. These three values is very important in the food preservation by the heat. Then first we will see the D value, and D value represent the decimal reduction time. So decimal reduction time is the time required at any temperature to destroy 90% of the spores or vegetative cells of a given organism. If 90% of the spores and vegetable cells is destroyed on a particular temperature, we are calling D value. So higher the temperature, the faster rate of destruction, and lower the temperature, the destruction power of the microorganism will be reduced. For example, of the D value, Clostridium sporangiens in given food at 120 degrees centigrade is 1 minute. And if you are decreasing the temperature, then you have to increase the time. So 115 degrees centigrade is 4 minutes. If you are decreasing further 110 degree, you have to increase the time. So 110 degrees centigrade is for the 10 minutes. The next is the Z value. So Z value is the number of degrees the temperature has to be increased in order to reduce the thermal death time tenfold. So Z value is relatively constant and depend on very little upon the environment. For spores, the bacteria, the Z value is used is 10 degree centigrade. So spore killing effect, the heat treatment can be expressed as a function of temperature and the time of the material has been extracted. So one example we'll take to understand the Z value. When 1 minute to kill 90% of the remaining spore at 120 degrees centigrade, it will take 10 minutes to obtain the same effect at 110 degrees centigrade, and it will take 100 degrees centigrade. Next is F value. Eating such a food at 111.1 degree for 2 minutes will have F value 0.2. So it means that one can obtain the same killing effect of spores and vegetative cells at low temperature, and provided the time of exposure is longer. So F value shows the heat treatment given to the food product to destroy bacteria. If our concern is the spore killing, then F is should be equal to 1, or it means that 1 minute at 121 degree centigrade we have to keep, or we can reduce the temperature for, and we can increase the time, or we can reduce further, and then or we can increase the time. So if we are increasing the temperature, we can reduce the time. If we are reducing the temperature, so that we have to increase the time to maintain the F value. And if F value will be achieved, so that we can easily kill the microorganism. Now some important books and food microbiology, fourth edition is there. Author is Martin Adams, Yuri Moss, Peter McCue. Second book, Modern Food Microbiology, Food Science Text Series. Author James M. J. Martin J. Losner, David A. Golden. Third book is Fundamental Food Microbiology. Author Vivek Ray, Arun Dunia. Fourth book is Food Microbiology and Introduction. Author is Carl Matthews, Calmia Kenya, Thomas Montevideo. And some important questions on the food microbiology. First question is, which of the following produce the citric acid? First one, Aspergillus. Second one, Pseudomonas. C option, Saccharomyces. D option, Clostridium. You have to select one correct answer. Then next question, which of the following organism is nitrogen fixing and found in rice field in symbiotic association with Azolla? Option A is Frankia. Option B, Polyphotrix. C is Spirulina. D is Anabina. Then you have to select again one correct answer. Which of the following is used as a biofertilizer for soybean crop? Option A is Rhizobium. Option B is Azotobacter. Option C is Nostoc. Option D is Azospirillum. Then next question. A common biopesticide for preventing plants from butterfly caterpillar is option A is Trichoderma, option B is Baculovirus, option C is Dumas, option D is Bacillus thuringiensis. Then you have to select any one correct answer. Next question. Yeast is used for the production of option A is tetracycline, option B is butanol, option C ethanol, option D is citric acid. Again, you have to select any correct answer. And thank you being with us. See you in the next class with a new topic. Thank you.